Everybody can see this, right?
Yes.
Yes.
Okay, let's talk about the muscle tissue. Chapter 9 is muscle tissue, and chapter 10 is the actual muscles. And there are certain muscles that I want you to remember, but right now we're going to talk about the muscle tissue itself. Basically, you're all muscle. The majority of your body is muscles. And muscles can transform chemical energy, which you know is ATP, into mechanical energy, resulting in force. We need to look at the types of muscle, the characteristics of muscle, and how muscles function. So, muscles are very different than other tissues, and they have their own terminology. Whenever you see the prefixes myo, mis, or sarco, we are referring to muscles. An example would be sarcoplasm. Instead of saying muscle cell cytoplasm, we are talking about sarcoplasm. Let me let that person in. So hang on 1 second. Let me let that person in since they're late. But Tyler, I'll let you in, and then we'll go from this. Okay? So it has its own terminology. It has its own prefixes. There are 3 types of muscle tissue, and you should know each one because I'm going to ask you questions on exam number 4 on the 3 types of muscles and how they differ. Skeletal, which is the majority of your muscles, cardiac, which obviously is your heart, and smooth. And we'll talk about where smooth muscle is. Okay? Basically, when you talk about skeletal and smooth muscles, these are the cells that we talk about when we talk about muscle fiber. We don't refer to the heart or cardiac muscle as having fiber. So let's talk about the most common muscle. One more student. Okay, I'll be nice and let you in. Okay. Let's go back. No more students can come in. Here we go. Okay, so skeletal muscle is basically the muscle that is attached to bone, that is attached to skin. These muscle fibers are the longest of all the muscle fibers, and they have stripes. And the stripes are called striations. They are basically voluntary muscles. And the word voluntary, as you know, can consciously be controlled. So our brain can control these muscles. All skeletal muscle is controlled by our brain. They contract rapidly, they easily tire, and they're very powerful. So the key words you should know about skeletal muscle is that they are striated and voluntary. Cardiac muscle is only found in the heart. It is also striped, so it is striated. But to differentiate it from skeletal muscle, it's involuntary. It cannot be controlled consciously. Okay? Yes, you can slow your heart rate down, but you can't stop it. Your heart has a steady rate due to its own pacemaker. So it's involuntary. It's going to beat whether you want it to or not. Okay? So the key words when we talk about cardiac muscle is that it is striated, just like skeletal muscle, but unlike skeletal muscle, it is involuntary. You cannot control it. Smooth muscle. Smooth muscle is just what it says. It has no striations. They have no stripes. That's why it's called smooth. It is found in the walls of hollow organs like your stomach, your urinary bladder, and your airways. It also is involuntary. You cannot control it. So the key words for smooth muscle are visceral, which means organs, non-striated, no stripes, and involuntary. You cannot control it. And this is a review. If you look at this review, okay? And you'll have that in your homework also. There are 4 major characteristics of all muscle. And they're in big, black, bold letters. So I'm going to ask you a question on this slide. Excitability. Excitability is the ability to receive and respond to stimuli, otherwise known as responsiveness. Contractility, the ability to shorten forcibly when stimulated. Extensibility, the ability to be stretched. And elasticity, the ability to recoil back to its original length. So know excitability, contractility, extensibility, and elasticity. Know those 4 characteristics of all muscle tissue. Okay. Muscles have important functions. You know that they produce movement. Not only movement in walking, but also movement of blood. They pump blood. The heart and also the smooth muscle of your arteries pump blood. Also, there are smooth muscles in your digestive tract which move food. So when you say they produce movement, it's not only walking, it's digestion of food and the propulsion of food from your mouth to your anus. And also they pump blood. They also maintain your posture and body position. They stabilize joints and they generate heat because they use ATP as they contract. It is an organ. Skeletal muscles are organs, and they're organs because they have different tissues that make them up. They have nerves, they have blood supply, they have connective tissue sheets, and they also are attached to other organs. Okay? Each muscle has its own nerve, its own artery, and its own vein. They rely on ATP. They rely on oxygen. They rely on glucose. And they also make waste. They also make waste. So contracting muscle fibers require huge amounts of oxygen and nutrients. Each muscle fiber is connected and covered by connective tissue. That connective tissue forms a sheath around the whole muscle and the individual muscles. It supports and reinforces the whole muscle. There are 3 sheaths that you should know. From outside to inside, the epimysium is a dense, irregular connective tissue surrounding the entire muscle. The perimysium is a connective tissue that surrounds groups of muscle fibers. This one is on the outside. This one is in the middle. And each muscle fiber is surrounded by a connective tissue called the endomysium. So know the difference between epi, peri, and endomesia. Here it is. Okay, here's the epimysium. It covers the entire muscle. Covers the entire muscle. Here is the perimysium. It covers a group of muscles. And here is the endomysium, which covers one fiber itself. Okay? Muscles are attached to bone. And basically, they're attached to bone at joints. They attach to bone in two different places. One is the insertion and one is the origin. The insertion is the attachment to movable bone. The origin is the attachment to immovable bone. So know the difference between insertion and origin. You don't have to know direct and indirect. That's not important. Know the two, insertion and origin. So let's now get into the nitty-gritty or the microanatomy of the muscle and the physiology of how a muscle contracts. Okay? Skeletal muscle is long, cylinder-like cells that contain multiple nuclei. The sarcolemma is the plasma membrane. The sarcoplasm is the muscle fiber cytoplasm. Inside of the sarcolemma, there are many glycosomes, bags of glycogen. Glycogen is stored in two places in your body. It is stored in muscles and it is stored in the liver. But we're now talking about muscles. Also inside muscles is a protein called myoglobin, muscle protein. And that muscle protein stores oxygen. So not only does the muscle store glycogen, it also stores oxygen. And inside the cytoplasm, there are organelles, just like there would be in any cell, but these cells are specialized. These organelles are special, and they are called, and we'll talk about each one individually, myofibrils, the sarcoplasmic reticulum, and T tubules. Myofibrils. Myofibrils are densely packed rods. A single muscle can contain thousands of them. And myofibrils account for 80% of the muscle cell volume. They have many features, and we'll talk about the striations, the sarcomeres, the myofilaments, and the molecular composition of myofilaments. This is a myofibril right here, a myofibril. And we'll talk more about the other things in the muscles. But the myofibrils are extremely important because it's only in muscle. The striations or stripes formed from repeating series of bands. And these bands are dark and light along the length of each myofibril. The dark regions are known as A bands. How do you remember that? Dark has an A in it, A bands. Inside the A band is an H zone. It's a lighter region in the middle of the A band. Within the H zone, there is an M line. The M line really stands for a protein, myomycin, that actually bisects the H zone vertically. Okay, I'm not going to really focus on H zone and M line. I'm going to focus on A bands. And I'm also going to focus on I bands. I bands are the lighter regions. How do I remember that? Because lighter has an I in it. So dark is A bands, lighter is I bands. But there is an important area in the I bands, and it's known as the Z disc. It is a coin-shaped sheet of proteins in the middle of the I band. So I want you to know from this slide striations, A bands, I bands, and Z bands. I'm not going to ask any questions on H zone or M line. Okay, but here's what we're talking about when we say dark and light. The A band is a dark band. The I band is a light band. And these are the striations. These are the striations. So please remember the dark bands are the A bands and the light bands are the I bands. The sarcomere, this should be also in bold. It is the functional unit of the muscle fiber. It contains an A band with half an I band at each end. It really is the distance between two Z discs. Individual sarcomeres are aligned end to end along the myofibril, just like a boxcar of a train. So the sarcomere is extremely important. Know that it is the functional unit of the muscle fiber. It contains one A band and half an I band, and the distance between sarcomeres is known as a Z disc. And I think here it is. Okay, here is the sarcomere. Look, it has an A band and half of an I band. Okay, a total A band and half of a I band. It is the distance between two Z discs. The sarcomere is the distance between two Z discs. Please know that. They also show you here the filaments, and we're going to talk about filaments in a second. But I want you to know that the thin filament is actin, and the thick filament is myosin. Those are the filaments that make up the myofibril. They are the structural filaments that make up the myofibril. Okay, and they're known as myofilaments. These are the structural filaments of the myofibril. The thin filaments are called actin. The thick filaments are called myosin. I don't really care if you know where they are, that's not important. Just know that the structural filaments are actin and myosin. And actin is the thin filament, and myosin is the thick filament. Here we have it. Here we have it. Here is the thin filament, which is known as actin. Here is the thick filament, known as myosin. And when the muscle contracts, these filaments are the reason the muscle contracts. Okay, we'll talk about the sliding filament theory of contraction. So basically, the thick filaments, which is the protein myosin, has a head and a tail. During contraction of a muscle, the head link thick and thin filaments together. So myosin and actin come together to form cross bridges. The thin filaments, which are composed of actin protein, come together and twist with the myosin filaments, forming a cross bridge. When they form this cross bridge, they attach to each other because of regulatory proteins. So far you have two proteins that you have to know: actin and myosin. Actin and myosin are the structural proteins of the myofibril. And they come together and form a cross bridge. They cannot come together without two other proteins being involved. And those two other proteins being involved are tropomyosin and troponin. They are known as the regulatory proteins. They are bound to actin. And when you want to contract your muscle, the regulatory proteins control the contraction and the cross bridging of actin and myosin. This is myosin. Myosin has a head and a tail. What happens in cross bridging is that the myosin head connects to actin. It's like a golf club. A golf club will have an opening in it, and it will connect to the actin heads. So the two heads connect. The myosin and the actin heads connect. But they cannot connect without the regulatory proteins. And the regulatory proteins are tropomyosin and troponin. What happens is that the regulatory proteins are on the structural protein actin. What happens is that these regulatory proteins turn the actin and make sure that the binding sites, these little holes, go into the golf club of the myosin heads, forming a cross bridge. So the forming of a cross bridge is the rotation of actin attaching to myosin because of the two regulatory proteins, and those regulatory proteins both begin with the letter T, troponin, and tropomyosin. Okay? Those are the big four proteins that I want you to know about the myofibril. They have other proteins, and there are two other proteins that you have to remember. And one of them is titin, Titin, another protein that begins with T. But titin is the elastic protein. It holds the thick filaments in place. It allows the muscle to stretch. It also allows the muscle to recoil, and it resists excessive stretching. There is also another protein in the myofilaments called dystrophin. And dystrophin links the thin filaments to the sarcolemma. Remember, the sarcolemma is the plasma membrane of muscles. Plasma membrane of muscles. There are other proteins. You see them here. You don't have to know. I want you to know 6 proteins: actin, myosin, structural proteins; troponin, tropomyosin, regulatory proteins; titin, the elastic protein, which helps the muscle recoil after stretching and resist excessive stretching; and dystrophin. Dystrophin is the protein that links the filaments to the plasma membrane known as the sarcolemma. You don't have to know the other proteins on this slide. Know the 6 proteins. Dystrophin is extremely important. Remember, it links the filaments to the plasma membrane or the sarcolemma. If there is a genetic disorder in that protein, dystrophin, you will have the most common and serious form of a muscular dystrophy, Duchenne muscular dystrophy. This is muscular dystrophy. It is a lack of the protein dystrophin, and it is a major muscle problem. Usually, these children are very, very ill and cannot use their muscles. Not only can they not use their skeletal muscles, they can't use their smooth muscles or their heart muscles. And without supportive care, people with Duchenne muscular dystrophy basically live longer than usual. They used to die in their 30s. Now they can live up to their 50s and 60s. So remember Duchenne muscular dystrophy, a genetic disorder involving the protein dystrophin. There it is. It causes a leakage, a tearing of the plasma membrane, allowing entry of excess calcium into the cell to damage the myofibrils. We will talk about calcium and why calcium is so important in muscle contraction. Okay, another interesting part of the muscle is the sarcoplasmic reticulum. Really, this is the endoplasmic reticulum of the muscle cell. It just has another name. Instead of endoplasmic, it's sarcoplasmic reticulum. Okay? The function is the most important. The function is it stores and releases calcium. Without calcium, your muscles will never contract, never. So the storage and release of calcium is the responsibility of the sarcoplasmic reticulum. Inside of the muscle cell, there is an extra organelle. This extra organelle is known as the T tubules. It is a tube formed by the protrusion of the muscle membrane, the sarcolemma, deep inside the cell. It allows electrical nerve transmissions to reach every single muscle fiber. And it forms a triad. And the triad is the area formed from the sarcomere, remember the functional part of the muscle, the T-tubule, and the termination of the T-tubule at the sarcomere. The relationship is extremely important because it acts as a voltage sensor and can change the shape in response to an electrical current. The membranes of the sarcoplasmic reticulum have proteins in it that control the opening of calcium channels. And remember, without calcium, your muscles cannot contract. So when an electrical impulse passes by, the T-tubules change shape, causing the sarcoplasmic reticular proteins to change shape, thus releasing calcium so the muscle can contract. And here is what we're talking about. Here's the triad. It is the T-tubules, the sarcoplasmic reticulum, two of them, all right? And here we have the muscle fibers themselves and the myofibrils. This is a myofibril. Remember, it is thousands of myofilaments, okay? So how does a muscle contract? Well, there is a theory, a sliding filament theory of contraction. The word contraction means that there is activation of cross bridges. Remember what cross bridges are. Cross bridges are the attachment of actin to myosin, regulated by troponin and tropomyosin. Contraction is the activation of cross bridges to generate force. Shortening occurs when tension is generated by cross bridges on the thin filaments that exceed the forces opposing shortening. What really happens is that the myosin filaments pull the actin filaments to shorten them, and then contraction ends when the cross bridges become inactive. The filament that moves in a contraction is the thin filaments or the actin filaments. In the relaxed state, the thick and thin filaments overlap only slightly. In the sliding filament model of contraction, during contraction, the thin filaments slide past the thick filaments, causing the actin and myosin to overlap more. In the filament model of contraction, the thin filaments move. They don't change shape, they overlap. When the nervous system stimulates the muscle fiber, the myosin heads are allowed to bind with actin, forming a cross bridge, which causes sliding, otherwise known as contraction. The cross bridges form and break several times, causing shortening of the muscle fiber. The Z disc are pulled toward the M line, the I bands shorten, the Z discs become closer, the H zones disappear, and the A bands move closer to each other. Here is a muscle fiber at rest. This is myosin. This is actin. This is a Z disc from here to here. This is an H zone. This is an I band. This is an A band, and this is an I band. When the muscle contracts, the H zone goes away. The I bands become closer to the A bands. The Z discs become closer to each other and shorten. And these form cross bridges. They form cross bridges. How do your muscles move? Well, your brain activates them. If you want to pick up something, the signal is transmitted down the spinal cord to the motor neurons, which then activate the muscle fibers. The neurons and muscle cells are all excitable cells, capable of producing an action potential, and these excitable cells are capable of changing the resting membrane potential. The action potential crosses from the neuron to the muscle cell via a neurotransmitter. Remember, we talked about chemical synapses. You have an electrical stimulus from the neuron giving you a neurotransmitter called acetylcholine in this case, which is now activating the muscle cell. That neurotransmitter activates the muscle cell, producing an electrical stimulus in the muscle to move. I'm not really concerned about ion channels. I'm only concerned about calcium. Okay, so don't worry about ion channels, but there are certain words that you really have to know. When the motor neurons meet the muscle fiber, this is called the neuromuscular junction. We know it as the motor end plate. Each muscle fiber has one neuromuscular junction and one motor neuron. So when the nervous system meets the muscle system, it's called the neuromuscular junction, otherwise known as a motor end plate. When the end of the axon terminal, and the muscle fiber meet, they are separated by a gel-filled space called the synaptic cleft. Inside the axon terminals, there are bags of neurotransmitter. These are called synaptic vesicles. These vesicles or bags contain the neurotransmitter acetylcholine. At the neuromuscular junction, you have the axon terminal from the neuron, the synaptic cleft, and the junctional folds of the sarcolemma, which is the muscle plasma membrane. And here it is. Here's the axon. This is a nerve. In the axon, you have synaptic vesicles, which contain the neurotransmitter. And then you have a synaptic cleft. And then you have the muscle. This is the neuromuscular junction, otherwise known as the motor end plate. Okay? So what really happens when you move your muscle, when your muscle contracts? Things happen at the neuromuscular junction. The muscle fiber is excited. There is an excitation, contraction, coupling, and there are cross bridges that are formed. Okay? So the action potential arrives at the axon terminal. We're still in the nerve. There are channels that open for calcium. Calcium enters the motor neuron. Calcium entry releases acetylcholine neurotransmitter into the synaptic cleft. The neurotransmitter, acetylcholine, diffuses across the cleft to get into the plasma membrane of the muscle, known as the sarcolemma. Acetylcholine binds to the receptors on the sarcolemma, opening the gates, allowing sodium to enter, resulting in electricity. To get rid of acetylcholine, there is an enzyme, and that enzyme is called acetylcholinesterase. It degrades acetylcholine in the synaptic cleft. And that's what happens at the synaptic cleft. Okay? That's just a review. All right? You know, there are many problems with neuromuscular transmission. One of the major diseases that we see in a neuromuscular disease problem is a very rare disease called myasthenia gravis. Now, you know that I was an ER doctor for 40 years. And for 40 years, I only saw 3 cases of myasthenia gravis. But it is hard to diagnose, but once you diagnose it, you can help the person with it. It is a disease characterized by muscle weakness, and the majority of muscle weakness is in the face. You have drooping of both eyelids. You have difficulty swallowing, difficulty talking, and generalized muscle weakness. The disease involves the shortage of acetylcholine receptors, because the acetylcholine receptors at the neuromuscular junction are being attacked by the person's antibodies. So therefore, it is an autoimmune disease, and it is a deadly disease unless it is treated and diagnosed. You certainly don't have to know much about polarization. I'm not going to ask you about polarization at the cleft or in the plasma membrane. That is not important. That is not important. Know that calcium is involved. We talked about that. Calcium is involved in neuromuscular transmission. And that's what's really important to understand. And understand about acetylcholine. Okay? But you have to know about excitation contraction coupling, known as EC coupling. It is the events that transmits the energy along the sarcolemma. That is known as excitation. When the sarcolemma is excited, it is coupled to the sliding of the myofilaments, and that's known as contraction. So the two together are known as excitation contraction coupling, also known as EC coupling. The energy is propagated along the sarcolemma and down the T tubules when the voltage sensitive proteins in the tubules stimulates the release of calcium from the sarcoplasmic reticulum. Calcium release leads to muscle contraction. And that is very important to understand. What happens if you don't have enough calcium? Well, if your body is low in calcium, that means that you can't form cross bridges. And when you can't form cross bridges is because the regulatory proteins can't move actin to troponin, to myosin. So tropomyosin and troponin are blocked, and you can't contract. So the muscle remains relaxed. So in the presence of low calcium, your muscles will all be relaxed. They cannot contract. If you have high calcium, say you have an endocrine disease and you have a high calcium, the calcium will bind to troponin. The troponin will change shape and move away from the tropomyosin, from the myosin and binding sites. The myosin heads are allowed to bind, forming a cross bridge. Cycling is initiated, causing the sarcomere to shorten, and then you will have muscle contraction. When the nervous stimulation ceases, calcium goes back into the sarcoplasmic reticulum and contraction ends. So the 4 steps of cross-bridge cycling is you need to form a cross bridge. And the formation of a cross bridge is that the myosin head attaches to the actin thin filament. You develop what we call a power stroke. The myosin head pivots and pulls actin towards the M line. The cross bridge is then detached. ATP attaches to the myosin head, causing the cross bridge to detach. Cocking of the myosin head, the energy from the making of ATP cocks the myosin head into a high energy state. This energy will be used for the power stroke in the next bridging cycle. So that's how you form cross bridges and a power stroke. It produces ATP to be used in the next cycle. So what happens when you die? Well, why do your muscles stiffen when you die? Your muscles stiffen between 3 and 4 hours after death. This is known as rigor mortis. Peak rigor mortis is 12 hours after you die. What happens is that intracellular calcium levels increase because ATP is no longer being synthesized. So the calcium can no longer be pumped back. And this results in cross bridge formation. Muscles will stay contracted until muscle proteins break down. This is how a pathologist can tell how long you have been dead within 12 hours of your death. Rigor mortis. Ooh, we're done. Are there any questions about muscle fibers?